So the tertiary structure of a polypeptide chain refers to the spatial arrangement of amino acids that are found far away from one another along the polypeptide chain. So the tertiary structure is simply the three-dimensional arrangement of the atoms, or the three-dimensional shape that the polypeptide chain will take within its local environment. And there are interactions that plays a role in creating and forming that tertiary structure of the polypeptide chain. So we have the hydrophobic interaction, disulfide bonds or disulfide bridge. We also have the hydrogen bonds and the ionic bonds. By far daw po, the most important factor or the driving force that forms a tertiary structure is the hydrophobic effect and the hydrophobic interaction. Kasi po, the majority of the proteins inside our bodies and inside our cells folds and create the tertiary structure in an aqueous solution, or in a solution where water is the dominant solvent molecules. So dito sa diagram, we have our proteins, and let's suppose that these red sections are the hydrophobic regions and these blue sections are the hydrophilic regions. So when we place them into a solution that contains water as a solvent, halimbawa may mga water molecules dito sa paligid nila. So yung blue section will tend to part on the surface or lie on that surface, while the red section will tend to aggregate at the center and form the core of that protein. So a protein in aqueous environment will have a hydrophobic core and hydrophilic surface. And the hydrophobic interaction is an intermolecular interaction. So what about intramolecular interaction? So we also have this specific type of intramolecular interaction or covalent bond that can also dictate tertiary structure, and these are known as disulfide bonds or disulfide bridge. So if we have two cysteine amino acids that are in close proximity, so we have two cysteine amino acids, and an oxidation reaction take place, we can form a covalent bond between our sulfurs, or between our sulfur groups. So basically, when oxidation reaction take place, we take away this H atom and this H atom. So we take two electrons, we take away two electrons, and we form the single covalent bonds between the sulfur. And this disulfide, this is a disulfide bonds or the disulfide bridge. So some proteins, especially those proteins that are destined to be in the extracellular environment, this polypeptide can be cross-linked via disulfide bonds between cysteine residue, and once we form the unit of these two cysteine, these cross-link units are called cysteines. Then we also have the hydrogen bonds. We have polar molecules on the side chains of those polar amino acids that can basically interact and form hydrogen bonds with the water molecules that are found in close proximity, and that can stabilize the structure of tertiary structure of our polypeptide. And finally, we can also have ionic interaction or ionic bonds. So di ba nga, there are certain amino acids that contain side chain that carry a positive charge, gaya ng lysine. Tapos meron din naman tayong acidic amino acids, di ba, that contains a negative charge naman on their side chain. So if we have the amino acids that contains a positive charge on their side chain in a close proximity with our amino acids that contains a negative charge on their side chain, and these two are close enough to form an ionic bond, and that ionic bond can also stabilize the tertiary structure of the polypeptide chain.